Hi, my name is Bianca Mulaney. I am Life Sciences, and this is my Bluesky proposal. The inspiration for my project came from my freshman seminar professor, whose main work is focused on biofilms. Biofilms are basically bacterial aggregates that form on surfaces. So this incorporates a wide range of bacteria and can form on nearly any surface. So the biofilms usually have a pretty big impact on a lot of real-world applications. For instance, biofilms can have beneficial effects because their resilience makes them useful in industry, and their relationship with plants contributes to plant prosperity. And in addition, they also contribute to the food chain as decomposers and producers. However, biofilms can also have a very detrimental effect. They actually are the #1 cause of health hazards in hospitals because they cause infection when they cover catheters or implants. They can also cover teeth, form biofilms over your teeth. They contribute to various diseases such as cystic fibrosis, and they can be an economic nuisance because they can cover oil pipes or contaminate water purification systems. So they're a really important problem to study, and the main reason why biofilms are so difficult to control is because once the bacteria that make up this biofilm actually form into a biofilm, they become part of an extracellular matrix. They actually grow an extracellular matrix that holds them together. And when you put the bacteria in this extracellular matrix, they undergo a physiological change that prevents them from being treated with conventional antibiotics that you would use to treat normal bacterial infections. So it's really important to be able to understand these interactions that are occurring between the bacteria and between the proteins and the polysaccharides and the nucleic acids that make up this extracellular matrix that are holding the bacteria together in order to somehow understand how biofilms assemble and disassemble. Because if you can understand that, you can address all of these problems that I mentioned previously. LS1a was really key for me to learn about the cellular interactions and what really occurs in cells on a microscopic level. For instance, in LS1a, we learned about lipid raft formation. And lipid rafts actually form on the cell membrane and can concentrate proteins in certain areas along the cell. And it's been found that in biofilms, lipid rafts actually attract certain proteins that hold the bacteria together. So if you could somehow disassemble the lipid rafts in bacterial cell membranes that are in biofilms, you could potentially prevent the proteins from binding to the lipid rafts and prevent the bacteria from binding to each other and cause the biofilm to disperse. It has been shown that when you add certain acids to the biofilm when it's assembled, you can actually disassemble the biofilm matrix by disassembling the lipid rafts in these bacterial cells. But you don't kill the bacteria. The bacteria are still alive even though they're dispersed into solution. They're not part of the biofilm. So my proposal is to somehow combine antibiotics with this acid lipid raft treatment. And if you had a combinatorial treatment of both antibiotics and the lipid raft treatment, you could both disperse the biofilm by dispersing the lipid rafts and kill the bacteria once they're dispersed.